Hello, thank you so much for having us. We're all very excited to be here. And after listening to the list of all these things you have coming up, I don't want to go home after this. My name's Amanda Patsis. I'm here with my colleagues David and Woonghee. And we're gonna talk to you a little bit today about all the things that are going on under your feet and in Soudan mine. It's sort of what the microbes there are doing. Has anyone here ever been to the mine? Most of you, yeah. Has anyone taken the historic tour? Awesome. So the site that we study, you probably drove past if you took that little cart on the tour. It's on the lowest level of the mine, the 27th level. And we work as sort of a collaborative team with all sorts of different types and disciplines of scientists to really think about what might be going on with the microbes, the rocks, and the water in this environment. And I wanted to right up front call out and thank Jim Essig, who is local here. I don't know, maybe some of you know him, but he is part of the DNR and really facilitates us being able to get access and get our science done in the mine. So we are so grateful for that. So I guess it's our pleasure today to introduce you to your microbial neighbors. Really what we think about in our science is how the minerals and rocks interact with the brine waters. So they're very saline, salty brine groundwaters that flow through the rocks in the mine. And the rocks and the brine impact each other, as well as impact the microbes and who is able to sort of live, survive, and thrive in the mine. So we'll sort of all talk about our own kind of chunk of this puzzle in the science that we do to understand how all of these different components are interacting. To sort of back up to our big overall research question, we're thinking about how microbes can make a living in fractured rock systems in this briny groundwater. So sort of on first look, it probably doesn't seem like a very friendly place inside a rock for a microbe to live, but they're making it work, right? So we're trying to understand how they do that. And folks study this in a lot of different places. What you're seeing here is the oceanic crust, right, on the ocean floor. Over here is the continental crust, which we're standing on right now. And it's pretty tough to sample and study microbes that are, say, a mile underground, right? We have to find some way to get to them. So in the ocean, we can sample hydrothermal vents using things like rovers. We can take cores, right, dig a big hole in ocean or continental crust. And especially on the surface, we can use existing infrastructure like the mine to sort of get access. So Soudan mine is, I think, a super special place for a lot of reasons, but through the lens of our research, it's this awesome window that we can use to look at and think about what these microbes so far underground are doing. Okay, so I'm gonna sort of take you on a journey of zooming in until we're thinking about things from a microbe's perspective. So to start, here are some rock cores that came out of the mine when they originally drilled, right? So these chunks of rock we can look at to understand the different types of rock that are in the mine. And we can also see, even with our visible eye, some areas like this that have fractures. And those fractures, which you'll learn more about, make a really good place for microbes to live. But if we really want to sort of think about what the microbes are doing, we need to be even more microscopic than things we can see with just our eye. So what you're looking at here on the left is this big purple structure is the tip of a needle. And if we zoom in on that tip, zoom in even more, each of these sort of orange Twinkies is one microbe. So when we're thinking about these, the size of these microbes, they're about 1 micrometer or micron, we call it, in length. So we can use that later to help you kind of see what scale we're thinking about. So this is a thin section, one slice of the rock, and this is from the cores that we were looking at earlier, right? So it's banded iron formation. You can kind of see some of those bands. And here we're zooming in even further to one tiny edge on one little fracture in this rock. And what you're seeing here is this scale bar represents 100 micrometers. So you could imagine like 100 microbes lined up end to end would be about this big, right? So from a microbe's perspective on that rock core, there's actually all of this surface that's available to them that they can kind of see and access. And they can get at these different types of minerals, things like pyrite and hematite, chlorite, and even some carbonate minerals. And they can use the things in that rock and the things that they're getting from the water around them to fuel their lives. So we'll talk a little bit more about sort of how all of that works. But first, I want to give everyone a second to take a breather, think about our new microbial perspective, and ask any questions you might have before we learn a little bit more about the fractures and water moving through them. Yeah.
Are you looking for things that have been there a long time?
Good question. Are we looking for things that have been there a long time? So ideally, yeah. And we've done some work to look at if the water that we're accessing in the lowest level of the mine is just rainwater from last week that has trickled down, or if it's been down there for a long time. And it seems to be very isolated and it looks different than meteoric or rainwater. And in some places of the Canadian Shield, this is work done in Canada rather than here, but they've estimated that the brines have been underground for thousands, hundreds of thousands of years. So the sort of hope is, and what we think is that we're accessing this really old water, microbes that have been separated from everything on the surface for a really long time. Oh, a lot of hands.
Yeah.
That's nice that you hear.
Whenever the glaciers are melting or people are going, oh, you're releasing microbes that, you know, shouldn't be released in the, so are you releasing microbes that shouldn't be released?
Good question. Okay, I have, I guess, a couple thoughts. The first would be, I think a lot of us think of microbes as like a scary and dangerous thing, right? Because there are some microbes that make you really sick. But in the grand scheme of things, there are so many types of microbes, it's less than 1% that can actually make you sick. And then I would say in the mine in particular, I think a lot of the microbes that can be harmful to humans have sort of evolved alongside of us and become good at infecting us. But the microbes have been isolated doing their own thing, so they never had the need to evolve to sort of infect humans. Does that make sense? Okay. How are we doing on time? Okay. Let's do one more question and we'll have lots more time.
What happens when the microbes come out of isolation?
So like when we bring them up to the surface?
Right, or when they ever come up to the surface on their own.
Yeah, so right, theoretically this water is underground, but at some point can, just like all of the groundwater that we like drink even, right? At some point it can reach rock that allows it to come back up to the surface. So actually a lot of folks will sample the microbes in those places because it's right, it sort of gives us an idea of who was down there. A lot of times these microbes can sort of live in those settings. For us, when we sort of outside of the natural order of things, take the microbes out of the ground, you'll hear from David, but we can actually like still get them to grow if we give them the right conditions in labs. Does that help answer your question? Okay. I'm gonna hand it off and we'll hear from Woonghee about some fractures, but you'll have lots more time for questions.
My name is Woonghee Lee, an international graduate student from South Korea, and I'm mostly studying on fractured rock and groundwater. So I'm going to talk about how we can access the microbes and how we can find the microbes in the mine. So this is the schematic diagram of the Soudan underground mine state park. So this is basically the geology under the state park over there. So there is our target boreholes around at the 27th levels of the mine, which is about 713.
Sorry.
Yeah, that'll be great. 730 meters down, then there is a shaft from the surface all the way through up to this 27th level, and there's the elevator working through the shaft. And this is the picture we took in front of the elevator. So this is me, this is Dave over there, and this is Chris, who recently graduated. So using this elevator, we can get there at the 27th level. So you can see there's a level number 27 and actually 2,341 feet below the surface and 800, maybe 88 feet below the sea level. So it's really deep from the surface. And there is our target borehole. And if we zoom in, you can see this is the shaft at the 27th level. And at the end of this way, we can find our 4 different target boreholes. And from this boreholes, we can observe this kind of biological activities. So there is the water flowing out from a borehole, and you can see there is a lot of iron precipitates around the borehole. It's all because of the microbial activities. And another video showing a bubble from the borehole. Maybe it's too tiny, but yeah, it's because the microbes making methane, the gas from the borehole. And these boreholes actually penetrate a fracture bed of aquifer. So these microbes are living in fractures and working to make this kind of interesting phenomena. So I will briefly explain what is the fracture bedrock aquifer and how the groundwater flows through this geologic layer. So this is the schematic diagram of the fracture bedrock aquifer. It's a kind of geologic layer containing water inside this fractured rock. And if we zoom in, we can find these fractures inside the rock, and the groundwater mostly flows through these fractures, which is because large open space compared to the really dense rock materials around there. And this groundwater delivers nutrients to microbes through fractures. So again, in this fracture bed of aquifer, we can see the groundwater mainly flows through these fractures. So if there is nutrients like this lead plume, groundwater mostly carries these nutrients through these fractures. You can see over there. And if we zoom in into the fractures, then we can see the microbes uptake the nutrient from the water, and also they interact with ambient rock matrix. So inside the Soudan mine, inside the borehole, we can see there is the active water rock micro interactions within fractures. So microbes mostly stay in fractures to take their nutrients for their metabolism, and also they have an active interaction with the ambient minerals. And Amanda and David will explain how these microbes work and how these microbes live inside the fractures. And before moving on there, I would like to take any questions if you have. Yes. I think you can choose.
Microbes. Are you studying them? What are you doing with these microbes? Are you studying them in a sense of discovery, or are you doing something with them?
Yes, we have. Yes, I guess the question is, what is my purpose for the studying? My focus is to understand how this groundwater supplies the nutrients to the microbes. So basically, it is kind of water microbe interaction. And as I mentioned, the microbes are doing many things inside the fractures, like a water mineral interactions or water or something interaction. But my focus is how the water effectively supplies the nutrients to microbes in the mine.
And I'll just add one quick thing. I'll talk more about like what we're doing to study independently those microbes and
what we're doing them in the lab and trying to find out about them. And so we'll talk about that when we get to my section. So yeah.
And some bigger picture,
And bigger
like what
picture.
we
Yeah.
can learn about.
yeah, thank
It's
you.
coming.
Wait, oh, we got more.
Yes.
This is so intriguing. I'm wondering if you're studying the secret of how the speed of water, flow of water, if that then impacts what kind of microbes can stay and hold on, as well as, access to them. If you could reveal those two.
That's a great question. Yes, actually, one of our interesting point is the flow rate of the groundwater, because the flow rate can affect the supply of the nutrients to the microbes. So we can assume that if the flow rate is really fast, then microbes may hard to uptake their nutrients because they will just quickly pass around. But if the flow rate is slow, they may have more time to take it. So that is one of the key questions we have.
On, stay still in one area.
Yeah, maybe Amanda and David are more expertise on that. They will explain about.
This is a little off topic, but how did you get interested in the and come all the way here from South Korea to study it?
Yes, I actually was a great question that reminds me why my original motivation, but I originally interested in water, and then I ended up to study the groundwater. And after studying this, I met this really nice project, and there was really nice, interesting research topic. So I found that there is one really nice scientific findings from there. So yeah, we are pursuing that, how the groundwater affects that kind of interesting biogeochemical reactions in there.
Thank you.
Thank you.
I'm back. All right. I will answer the question about how do microbes stick around, because I love that question. What you're looking at right here actually is a little bit fuzzy and hard to see, but it's right here's that scale bar again. This time these little, if you can see the kind of like little logs here, those are individual microbes, and this is actually a sample from Sudan that we were able to image. So a lot of these microbes you can see are surrounded by, the big chunks are minerals, but there's also this kind of sticky looking stuff, and that's part of a biofilm. So that's also what's being shown over here, that yellow tan is representing sticky biofilm, and then there are all those colorful microbes in it. But a lot of times if microbes find somewhere that's like, ooh, yeah, this is a great place to live and I want to stick around, they'll make this sticky substance and sort of, as a community, like grab onto the rocks or that place in the fracture. Sort of like if you imagine like at the beach, sometimes you get those really slimy rocks. It's sort of like that. In this case, microbes are making them.
They make communities.
I'm going to talk a little bit about that. So I know you met me before, but I was going to just introduce the type of research that I do. So I primarily do microbial ecology. I think a lot about how microbes interact with each other and with their environment. And to do that, I use their DNA. So in the mine, we can sort of filter water, push it through this really fine filter and capture microbes. And then I take them back to the lab, pop them open and isolate their DNA, and then sequence that DNA. And by looking at which genes they have and which proteins they can make, we can learn more about what they are able to do, how are they able to survive. So for our story today, I'm going to focus on how are they making their energy to survive, which is one of the things we've been able to learn about. So how are the microbes in Sudan getting energy? All they've really got is rocks and this brine, right? So they're actually using the same process that every other living thing does. It's called respiration. And that's more than sort of, we might think of breathing air in and out of our lungs. But when I'm using respiration here, it's more the whole chemical reaction, the chain of processes that go from you eating something, breathing, and turning those things into energy. So what does that look like in humans? Well, you're all enjoying a very lovely lunch right now. And once you digest that lunch, your cell will take electrons off of the burger that you're eating, and it will move them onto the oxygen that you breathe. And through that process of moving electrons, your cells are able to capture energy, and then you use that energy to all the other processes that they need to power us moving and breathing, right? So in humans, it looks like this. Even in some microbes that are able to breathe oxygen, it looks like this. But if you think about microbes living in Sudan, hamburgers are not that easy to come by. And we know that there's not a lot of oxygen. So the waters in the ground there are what we call anoxic, which means there's no oxygen around for them to breathe. So instead, they get a little creative and they eat and breathe different chemicals that they can get from the rocks. So these are just some examples, the things that they eat. We call electron donors, but all of these different compounds that might be iron or nitrogen or sulfur, they will pull an electron from those. This is their hamburger, and they'll put that electron onto the things they breathe, which we call electron acceptors. This can be different forms of those same iron, sulfur, nitrogen, right? So rather than eating hamburgers, breathing oxygen like we do, the microbes in the mine have special proteins that let them carry out these pathways to eat different metals or chemical compounds, elements, right? And breathe things that they can get from the rocks to make their energy. Even with this way to make energy, it's not you don't get the most out of it. It's no hamburger. So there's a couple strategies that the microbes in the mine have evolved to help them really make that energy last as long as it can. And the first of those strategies I called here, just take it easy. They sort of just float and hang out and spend the bare minimum of energy on things they absolutely have to do to survive. And otherwise, they just kind of let life happen. So in a lot of cases, these microbes can take weeks, months, years, in some cases, like thousands of years are the estimates for how long before the cells divide. So these microbes are hanging out underground, conserving their energy as much as possible for a really long time. And another way that they will sort of conserve their energy, much like myself in the winter, they can become a dormant spore. They build a really strong coat around them and become essentially inactive, almost unalive. And then one day the conditions are right and they can sense that and it triggers them to reawaken. So using these strategies, they can sort of make their not quite a hamburger chemicals last as long as possible so that they can survive through tougher times. And another way that they sort of make this energy last, which comes back to your question, is through the magical power of friendship, right? These microbial community members will sort of depend on their neighbors for different things. Rather than being able to carry out a whole process all by themselves, having to maintain all the proteins to do that, some of the microbes will do the first couple steps and then they'll hand it off. And the next microbe will do maybe the last couple steps. And by the end, as a community, they have that important thing they need and they can share it back, but they're each taking on only a little bit of that burden. So it's kind of like, right, asking your neighbor for sugar or borrowing someone's oven if you don't have one. And sort of in that way, they can work together, and even though they've got a little bit of energy available, it's enough. Okay, another break for you all. Does anyone have questions?
I have a million questions.
Would these microbes be, would they fit the definition of bacteria?
Yeah, lots of them are bacteria, and then there's another branch of life called archaea, and we see some of those too. But yeah.
So I did a little bit of reading in I think anaerobic bacteria.
Mhm.
They're poisoned by oxygen. Is that right?
Yeah,
They
so
can't
there,
live up here.
yeah, there are a couple different types. So they're called obligate anaerobes, which means if they see oxygen, it's a bad day and they're not going to make it. And then there are some that they maybe don't like oxygen, but if there's a little around, they can deal.
Do they have any relationship to chlorophyll at all? And that's totally related.
Ooh, yeah, good question. So on the surface, right, chlorophyll is a great way to make energy. This is how plants and algae photosynthesize, but there's no sunlight in the subsurface. They don't have chlorophyll or a way to make energy in that way, which is why they're stuck eating things they get from the rock. Good question. Gonna let me know when it's time.
Can you tell how old the microbes are?
Ooh, really good question. So for like an individual microbe, I would say there's not a great way to say, this guy was born a year ago. But one thing that we do is think about the evolutionary history of microbes. So for different like species or types of microbes, we can sort of see when they evolved and when they branched. And we do see that a lot of the microbes living underground and living in Sudan branched really early on in the history of life. So evolutionarily, they're very old, and they were sort of what some of the earlier life would have looked like. Does that help?
Thank you.
Yeah.
Ms. Donna? This doesn't seem possible from what you said, but does it have any connection to bats? Have you noticed any
Ooh.
of them on the bats or?
Yeah, great question. So the microbes that we're finding don't have much of a connection with bats, but there's like a white fungus, right, that can hurt the bats. That's a different type of microbe, not one that we find in the rock below the mine, but certainly one that exists in the mine, and it's not great for the bats there.
Back here. How are microbes born and how do they die and what happens to that?
Good question. I love this. Okay. Microbes, essentially one microbial cell can divide and split into two. And there are some ways that microbes can kind of share their DNA, which would sort of be like our equivalent, right, of an egg and sperm coming together. But for the most part, a microbe can just decide to be two. And now there are two daughter cells that look just like the parent. As for how they die, there's lots of different ways. A big thing in these systems is viruses, which we haven't looked at yet. I would love to look at the DNA of viruses, but they can cause the cells to die and pop. And that's a really big part of sort of recycling nutrients and allowing the microbe and the whole community to survive in these really nutrient limited settings.
We have a question here.
In our wetlands, we have these microbes that maybe they run off of oxygen, but in the low oxygen environment, they'll somehow switch over to a sulfur process and it ends up they're pulling the oxygen off of the sulfide and you're killing wild rice. And I'm curious, in the mine, are you getting these microbes that can switch from oxygen to a sulfur? I see you had a slide what they're using. Are they using sulfur in the mine the way our microbes in our wetland are using sulfur to kill our wild rice?
Yeah, absolutely. So we see microbes both in wetlands and in the mine that can use some forms of sulfur to sort of eat and then use other forms of sulfur to breathe, right? And they'll sort of, through that process, it will change sulfate to sulfite and sulfide, right? Which some forms of that are, well, better or worse, really bad for wild rice. So we do see microbes that can do that, just like in the wetlands. And something that I'm interested in that we'll maybe hear a little bit about at the end is the idea of bioremediation. Is there anything that the microbes can help us do in areas that have too much sulfur? Can they turn it into something really bad, from something really bad into something a little better, or into something we can more easily take out of the water? So there's lots of other folks that do research on those types of things, but it's definitely sort of one of the more practical applications of what we think about. Okay, I'm gonna pass it over to David.
No, it should be fine. Okay. All right, hello. All right, so we're gonna move,
Oh, we're going to move on to some of my work. I'm also a grad student as part of this project, and my focus is microbial physiology. So I study the bacteria itself and kind of understand exactly what they do, not always in a community setting, but also in the lab itself. And so kind of working with one individual microbe that we find, and I'll be talking a little about the story of one of the microbes that we found in the mine, and we can work with it in the lab. We've isolated it and characterized it. We gave it a name. And so we'll talk a little bit about that. So I do work both in the mine, but also in the lab to help study these organisms. Okay, so we talked a little bit, or Amanda already talked a little bit about this kind of process of how they make their energy kind of by breathing different kind of compounds and eating different types of food. But the one that our lab particularly studies is how do they basically breathe rocks or metals. So when we think about iron in kind of its most common form that we see out here in kind of the whole iron range, we think of kind of solid rocks, right? And so if we think of a big rock and trying to breathe it, it doesn't really make sense. Oxygen, we can bring it in, we could bring that into our body, but it's pretty hard for a little tiny bacteria to bring a rock inside of itself, right? And so how do they do this process of getting an electron from their food source onto this big rock, right? And so, wait, actually, so this is kind of a little demo thing that I like to use, but this is a actually piece of banded iron formation that we have from the mines, and it is really high density iron, and so because of that, it is actually kind of magnetic. Oh, thank you so much. And so we have these little bacteria that are itty bitty and tiny, and they have to breathe this big rock, right? And it makes it really difficult for them. These are just little like 3D printed kind of fake bacteria that have little magnets on them. I also made a bunch of these that we can hand out today if people are interested in having these little kind of toy bacteria. Thank you. But yeah, and so the big question is, how do they do this process, right? How do they interact with this? And so the real answer is they can't. They can't bring the rock inside of them, and so they have to get the electron outside of themselves. And this is a process we're really interested in studying, and because these are a very specific group of organisms that a lot of bacteria can't do, of getting the electron from inside the cell to outside the cell. And there are a lot of applications for why we're interested in this that I'll talk about at the end. But this is kind of the story of how we found one of these in the mine and why we're interested in studying. And so how do we find these iron breathers? Really what this means is that we go in and we collect a lot of sample. This is a picture of one of our collaborators, Scott Alexander, who's working in one of our boreholes, and we're basically just pumping material out. So this is us pumping out a lot of that brine water, collecting sample in particulate. And so this is what the brine looks like if we collect it deep enough, is that you can see kind of like this salt formation at the bottom inside that water because it's really, really salty water. But there's also this kind of like black particulate that's floating around, and that is one of those iron minerals that we think the bacteria are interacting with. And so they're going to be hanging out on these rocks. At least that's what we thought, or now we know. And so what we do is we just basically take some of this black kind of particulate or this mineral, we put it into certain types of media. And so these are just these little test tubes that we make that have very specific components in them that gives them a little bit of food. They're very specific conditions that allow just these organisms to grow. So we can select for the ones that we want and make sure that we basically grow the ones that we're interested in and not anything else. And we just kind of put them in there and let them sit and do their thing. It turns out organisms from the deep underground, that have been around for a long time grow very, very slowly because they've had, millions of years to like do their thing very slowly. And so we basically just sit there and we wait. And so we just take care of them, we read to them, we give them some love, and then we kind of just grow them up and hopefully something grows, right? And so we just make sure they're happy. And eventually, if we do this long enough and we give them the right conditions and we have the right bugs, they will actually start to grow and they'll start to do things. And so this is kind of a time course. This is over several months. And what we can see is that the iron that we started with has now changed. So as the bacteria is breathing the iron that's in there, it's actually changing the properties of it and turning it into a different form that potentially some other one of their neighbors could use also. But in our condition, we're just kind of isolating that process. And so as that iron changes, it goes from this kind of the classic orangey kind of irony color to something that's actually completely clear, and it's more of a particulate as well, right? And so what we can do then is after we've grown a bunch of this, we can grow this onto solid media or basically just plates. We can spread them out and we can try to isolate, which is a term we use of basically picking just a single one. And eventually we just get this one little red dot. And that red dot is actually just a bunch of bacteria of one type that we want to study and we know can do this process. And that reddish pinkish color comes from the proteins it uses in order to basically do the process of breathing the rock. And so this is something that we're interested in. And this is that one kind of organism that we pull from the mines that we got to characterize. I also got to name it. Oh, I clicked the wrong button. Right. And sorry, what was that? Oh, that's a great question. So the plate itself is going to be this orangey color, and that's why the top part is that kind of an orangish color. And as there's more bacteria around, it's going to start to change it and become clear at the bottom. And all the white stuff is just salt. So we try to mimic the conditions that are from the mine itself. The mine water down there is super, super salty. Yeah, it's 3 times saltier than the ocean. And so because of that, if you let these plates sit long enough, the salt starts to come out. So that's actually just the salt precipitating over time. Very good question. Thank you. Okay, and so this is the organism that we isolated, and I named it Metalumcola vericulae. It's just Latin, and we got to pick a fun name. And it basically means that this is an inhabitant of a mine, right? Because we found in a mine that works with the iron and puts a little bit of force or pressure on it. And so we went through the whole process of naming it. And so it's a very unique organism and that we don't find anywhere else on the planet. Its closest neighbor is very, very distant and is like found in like a marsh in Japan. And that's very distant. And so it's really unique and really interesting. And when we look at it under a microscope, it's this itty bitty little guy. They just have these little rods. And one thing you'll notice is that in kind of the bottom right picture is that some of them have this little circle at the end. Those are those spores that Amanda was talking about earlier. So it actually has that kind of process of creating spores and being able to kind of survive and persist inside the environment and just become dormant. And so this is something that we physically have seen and we have proof of now. And so we're in the process of studying this organism and learning more about it. And this is a really complicated slide, but this is all the machinery it needs to get that electron from inside to outside to breathe that rock. And these are 2 different organisms. So the left panel in the middle are all really well understood organisms and how they are able to do it. And on the right side is our bacteria, which is from the mine that we have no clue how it does that last step. That's why there's a big question mark there. And so we're really excited to find out this new strategy or either potentially really old strategy, because this is from deep underground, of a way to do this process. And so that's why we're interested in it. And kind of the last, I mean, maybe not the last question, but one of the big things that we get asked is, why do we care about these organisms? Why do we study them? This kind of gets at some of your questions. So if we can study a bacteria that take an electron from inside the cell and put it on a rock, can we do the same process of getting that electron from inside the cell, but instead put it onto something like an electrode or a circuit or something like that, and have these bacteria actually produce a little bit of electricity for us? And this is what we're able to do, and this is something that our lab studies, is that we take a bunch of bacteria, so this is what we call a reactor, where there's a bunch of bacteria inside of this. This is why it's kind of a pinkish color. We have these plates here that are capturing the electricity, and what we have here is a graph over time that every time that we add a little bit of food, so that's the donor, or like that hamburger in this case, they will eat that up, and then the current production, so the amount of electricity it produces, goes up. Once it runs out of hamburgers, it goes down. We add a little bit more hamburger, and it goes back up and down, up and down. And this is something that we can find from organisms in the mine, but also all over the planet. And this is why we are interested in studying this specifically. And then kind of going on to a bigger picture and kind of wrapping up our whole story here. These are kind of all the major reasons why we're interested in studying all the microbes in Sudan. But these are kind of all potential aspects of what our research kind of can answer. First, of course, is the microbial neighbors. What is in your backyard? What are they doing? How are they surviving? How have they been there for so long? Bioremediation, as Amanda said, using these organisms to get rid of some of the really bad stuff, right? Because we know they can break down iron, they can break down some other heavy metals or other products like the sulfur, and so we're interested in that aspect of it. Also, you know, the layer of the rock record that we are in dates at 2.7 billion years old, roughly. Really, really, really old. Predates a lot of things on the planet. And so we're interested in what, you know, bacteria back then could have been like. If they've been trapped there that long, or is this kind of a kind of descendants from those things, and so on and so forth. So we're interested in that. And then the last thing is, well, you know, you're in a really salty place, very like kind of weird conditions. There's a lot of iron around. What are other environments that are like that? And one of the big ones is Mars is like that, right? And so if we can study these organisms and understand how they survive, could that also be applied to other planets that are also similar? And so we also asked that question as well. Okay, and I think that, last thing we just want to acknowledge once again, everyone on the Banded Together project. So it's not just us 3 grad students. There's a lot of professors that are involved, other grad students that are involved across both our university, UMD, and then also Lehigh University. And then we also want to thank Jim Essig again, and then also all the people at the Parks Department. And with that, we will take questions. Sorry, I kind of blew through that. Yeah.
Since the rocks were hot at one time in the past,
Yes.
and there were no microbes there deep in the earth, how did they get there?
So that's a great question. So there was a point where, okay, this is going to get into a little bit of history. The layer of the rock record that we're in at one point actually wasn't that deep down. So basically on the Canadian Shield, which is where we are, some of the plates actually shifted under. And so the layer that we are was actually used to be at the bottom of the ocean is what we think. And so some of the microbes there might have been trapped as the plates shifted and got caught under there. That's like one of the theories we have, but you also just get, because of all the fracture network in the water, as long as water can get down there, microbes can get down there. So there are other hypotheses also, but great question.
Like a Rusty and then Bill.
I hope I can express this in a way that makes sense. Amanda told us that they reproduce by dividing, and that the 2 produce sister cells are alike. But I assume there's always the possibility of mutation.
Yep.
So if there's a possibility of mutation, there's a possibility of evolution, change. So then these things have a very slow life cycle. Given that, do you have any way to measure changes in that?
Oh, that's a great question.
Or is that too broad a period of time?
Oh, no, that's a really, really good question. And we would love to see that change over that historic amount of time, but you're right in the sense that it is broad because it is so slow. It makes it harder to track that. I mean, we've only been studying this for, a couple years, and they've been around for billions of years. And so tracking all those changes over time is just not possible. What we can do instead is, if we get back to the DNA part that Amanda said, is if we look at kind of all the members that are there and track all the changes between siblings, right? You said that, even though they're identical, like siblings wise, every time they replicate or they divide, if one mutation happens between the sibling, eventually it spreads. And so by tracking all the changes of the whole population, we can get an idea of it. And the more changes we see, the more like further away they are from each other. That's one way we could get around that question, but it's not as precise, and there is a little bit of room for questions and error in that. But that's, yeah, great question.
I guess I have a 2-part question. One is this organism that you've named found anywhere else that you know of? And then secondly, are the geology people tagged on to your work because of the FE2, FE3 change in terms of, in that mine, you've got BIF that's magnetite and BIF that's hematite.
Mhm.
So is there some aspect of explaining the geology that resulted in the iron formation on the Vermilion Range
Right.
from these microbes?
Yeah, of course. Great questions. So for the first part, when we look at kind of all the microbes that have been described, so looking at kind of all the DNA that we've ever extracted for every microbe on the planet, we don't see anything that's that similar to this. There is like one specific gene that we use as a marker to kind of figure out how closely things are related. And even then, we're looking at like, they're only like about 92% identical, which is vastly different. Yeah, this would be like extremely far distances. Yeah, and so we don't see anything exactly like it. There are other organisms that are found on the bottom of the ocean in other places. So, off the coast of Japan, there are some things like in some weird swamps out there that are somewhat related, but that's, when we say related, it's very loosely. So it's very different from anything else that's been described, and that's why we're really excited. Your other question about like, I work with geologists and kind of understand this. There is another grad student who recently he finished and is now working somewhere else, who I worked a lot with, and we've kind of he was mostly the hard rock geologist of our group, and so he was the one that was kind of characterizing all the rocks and minerals, and we talked a lot about the role that this organism plays, but also other ones in those formations. So he's described a lot of like potential aspects that could have formed some of the minerals that we see, yeah, so absolutely, we work together and we talk a lot about that. And if you want to know more, we can chat after.
Why is the water salty? Is it sodium chloride or is it something else?
Amazing questions. So why it's salty, we have a couple debates on. One is that rock or the water has been interacting with the rock and the mineral down there for so long that it just kind of strips away some of the salt and stuff from the rock, and that could build up that way. Another way we think about it is that if this is just ancient, ocean water that has been trapped down there, it'll concentrate over time. So those are all kind of hypotheses we have. It is not sodium chloride dominant, so it actually is mostly calcium chloride, which is different from the ocean. So our ocean is mostly sodium chloride, but the water we find down there is actually like very different salt composition. So that's actually a really good point, and it tells us that water. That's one of the ways we can tell that our water's pretty separated from meteoric or rainwater. It tells us that there is separation there, and it takes a long time for that to interact. And so that's actually one way we know that they keep to themselves. Great question.
Non-scientific, I have two questions.
Sure.
The other one, quote unquote, more important, but just watching and listening to you guys, I would love to do a scientific experiment of having you three sit in front of elementary school kids, middle high school kids, and then have someone else do, their grade level science presentation. And I bet afterwards, those kids listening to you, even if they don't know anything, are gonna feel so energized. They want to study microbiology and everything. It's so awesome.
Oh, thank you. Yeah. Thank you. Yeah.
I'd go out and sign for a course right now.
Yeah.
A serious question though, because I'm into language. We use breathing all the time.
Sure.
Listen, breathing to me is air.
Mhm.
Eating poop and pee, solids and liquids.
Sure.
Is there a different word you can use rather than breathing? And
Absolutely.
could you please spell it for me?
Oh, so great questions. First, there are actually a lot of people who actually study science education, and so there actually are a lot of people who are doing exactly that, of interacting with younger and younger students in order to get them interested in science earlier. That's like a whole field of science. Yeah. And then for the second question, breathing is not the word that we most commonly use. Usually we say respire. So in the case of respiration, the reason that we use that terminology and why you probably think of gases and stuff is that for humans, that process or what we're doing with oxygen is chemically the same as what these bacteria are doing with these other compounds. And so what we're doing energetically is we're also giving away our electrons to the oxygen we're breathing, and then it gets sent off as carbon dioxide. And so that process, the chemical process is identical. It just so happens that for humans and for most of life on the planet, they use oxygen. And so it's more of which word comes first or what you use what for. It's just that the common perception would be, that breathing or respiration is only for gases. If we want to talk it very nitty gritty, sometimes we talk about it just as electron acceptor and donor, right? And so, so we would use oxidation and reduction, and that becomes the chemical level of just moving electron states. So we call those redox reactions, but at the end of the day, we still talk about it as respiring. Yeah, so there's no one fancy Latin word, sorry.
I want to commend, how engaging you all are and how knowledgeable you are. But I also just feel so proud to be a part of a community that can really hold its own in this conversation. And
Yeah, great question.
Such phenomenal question. I am not much a scientist, but I am a poet, and I'm wondering, when you do this work, what are you thinking about? What are you learning about the relationship between water and life?
Oh, wow. That gets real deep, I guess. No, that's great. I mean, so all life does need water. That is a given, right? Our bodies are 70%, I think, water. And it's just that water is required for a lot of these, even the most basic chemical reactions that kind of govern everything that happens in our body. And so water is absolutely essential. You really don't find life without water. There are some places where you get very, very dry and you can still find microbes, but they aren't active as long as there's no water around. So they'll do those ththings where they go dormant and they go quiet, but they won't do any of these processes without water. And so this is, that's the very scientific answer. But it really is just that water is absolutely essential. This is why we always look for water on Mars or on other planets, because without that water, we don't think life can exist. So yeah.
Follow the water to find the microbes.
Exactly. Yeah.
And what are there evolutionary lessons that are relevant or related to humans, you know, I understand about. I guess I'm just so fascinated on the sort of like travel back in time that you get to do through this process. I'm just curious to linger there for a second, if you have any takeaways that you find interesting about that.
That's a good question. I would say that, so when we track like microbes and the kind of the history of that, and we also do this with organisms that kind of are commensal or kind of grow around your body, not always harmful ones. There's a lot of like healthy microbes that are in your body that are helping you eat your hamburger or digesting it right now. It turns out your body can't break down everything in a hamburger as well. And so they're doing the same thing in your body to help you get down to the basic things that you can eat and consume. And so, you know, like I would say that are the big takeaways that like microbes are essential for all the processes on the planet, or most of them. And so like we've been around and been with microbes for a long time. I know there's a really strong, you know, negative connotation because we only ever think about that tiny percent that are harmful. But really we're learning more and more about the good ones in your body and on the planet and in the soil and in the dirt and all the amazing things that they can do and how we can leverage them and use them. And so I would say the big takeaway is just that like not all microbes are bad.
Thought on that.
Oh yeah.
Maybe we can pull this up at the end, but one thing, I do a lot of work with like evolution. So thinking about how microbes are related and also like working backwards in time to understand when they started to become different. So I think, you know, on the one hand, part of it is just this idea that like we, which you maybe feel are so different than a microbe, can sort of track back, right? A lot of the different like proteins and things that do that process of respiration that we talked about, it's the exact same DNA with the exact same code that is in your body and in the microbe's body, right? So that's always pretty crazy for me to think about, that in the grand scheme of things, right, everything is really not that different. And then the other part of that, maybe I can pull up the sort of one of the more recent trees of life. It's sort of this giant tree with all these branches, and they're almost all microbes. And then there's this little tiny one that has, you know, plants, animals, everything, just like a little asterisk in the corner. So sometimes I think taking the really big picture look at evolution can sort of put things in perspective for us.
That's what I'm talking about. That H2O molecule that made us movie really messed me up.
I just like to do this active for all of us, because the National Science Foundation is like 8 to 10 billion a year. And it's really important to fund this research. To give you an example, there's a chap by the name of Terry Mullins, who went to Yellowstone National Park and looked at these thermal hot springs and found a little bug, a thermophilic autologous organism, and developed something called polymerase chain reaction, which modernized the whole field of molecular biology in the 1980s. He's now a surfer after getting his Nobel Prize. And that led to the sequencing of the Human Genome Project with about 4.5 billion nucleotides around 2000 to 2005 or so. And then that led to what's called the Human Microbiome Project from the National Institutes of Health, which is about 40 billion of your taxpayer dollars. That's all of you in here. About 200 million goes to the Human Microbiome Project, and that looks at these bugs and health and disease. We have about 39 trillion, that's a T, microorganisms on each one of our bodies on about 30 trillion cells. And the microbiome may be disrupted in certain individuals, leading to what we call chronic inflammation and looking at inflammatory disorders, and also something called cancer. So there's relationships between the microbiome dysbiosis and these entities. So it's important to put this in context, context, in that iron anaerobic organisms may be very important. You may stumble onto something very useful, particularly in bioremediation, like getting rid of oil and oil spills and things like that, and dispersing that. So this is a very important topic. Thank you.
No, thank you, yeah.
Dr. Rom, I was just gonna say the same thing.
I'm just gonna add a little bit in relation to evolution and to the word respiration in the human being. We all have little furnaces in almost all of our cells called mitochondria. And that is where the electron transport happens. That's where we create our energy. And that electron transport is where the respiration happens. So at some point, bacteria became a part of us and are responsible for creating our energy. And so oxygen gets delivered to the mitochondria, fats and carbohydrates, sugars get delivered to the mitochondria, and the electron transport chain happens. So respiration, as another doctor, respiration is not the breathing in of oxygen, it's the end product of the electron transport chain that involves oxygen, which is a bacteria that we took in.
Thank you. I love hanging out with these doctors. Internet, or is this one of your own?
No, this is my own.
Yes.
Yes. So you talked about how there are some avenues of mutation and how it seems like they reproduce asexually most of the time. Is it possible that they produce sexually? And if so, what are the chances of mutation through that?
Okay, so good question. So most bacteria, I should say, don't really have like what we would say is like the traditional sexual kind of reproduction where they kind of like pair chromosomes and split DNA that way. There are processes to move DNA, what we say laterally or horizontally. So instead of like, from mother to daughter, which would be vertically, they go kind of side to side. There are kind of mobile gene elements that exist. So viruses can do this, plasmid or other things that we call plasmids, can jump from organism to organism. That is very possible. And in that case, they would be incorporating kind of all of the information there instead of like single mutations that we think of that can naturally occur. And so the jumping of genetic material like side to side happens pretty frequently and pretty readily, but that all comes from the same kind of pool of genetic information that was already there. And so like the chances of spawning something brand new is unlikely through this process. And so like something like crazy change of them to adapt to a human or something like that would be unlikely or almost impossible. And so when we're talking about like that mutation, it's usually very slow and very more adapted to what their lifestyle currently is. And so there were some questions about like if there's a chance they can get out and whatever. The organisms have been living and kind of adapted to this anaerobic, super salty, no food, no nothing kind of environment, grow really slowly. It would be incredibly difficult for them to evolve to a point where they can even survive in oxygen, and then on top of that survive in our bodies or survive on anything on the surface. And so like when I work with these organisms, I have to keep them in sealed tubes that don't have any oxygen in them. They have to be grown at different temperatures with really weird food, a lot of salt. It's basically really difficult to get them to crosstalk and stuff like that.
Thank you.
Yeah, well, just real quick, thank you for having us. Also, if you want like these little bacteria magnets coming up, I have some for everybody.
Probably the rocks you find in your backyard, but it's pretty fun to play with magnets on them. So
yeah. Oh, here, I don't know. I'll put them out somewhere. Oh, here.
I'll take them.
Don't worry.
Oh, okay.
Are you guys having a chance to enjoy the culture and healing?
Okay.
today,
I work with my grandson.
I mean, oh.
I have 3 grandsons. Oh, no.
Thank you.
all that time
Here you
you've been coming up